students, in this video we are going to recall the structure of an antibody molecule along with the case-based question. So the question is identify A, D, E, F in the diagram of an antibody molecule given. So let us first recall the structure of an antibody molecule. We know that B lymphocytes. So B lymphocytes will be producing the antibody molecules. These are called as an army of proteins. Chemically they are glycoproteins. Only B lymphocytes will be producing these army of proteins. So T lymphocytes will be helping the B lymphocytes to produce the proteins. T lymphocytes cannot produce antibodies. This is point number 1. Second one is that, coming to the structure, the antibody molecule consists of 4 polypeptide chains. 4 polypeptide chains will be present. Okay. So 2 chains are smaller chains which are called as light chains. So this one, small chain number 1, light chain, and this is small chain or light chain number 2. So 2 light chains are present. So we represent that as L2. Okay, and other 2 chains are longer chains and those are called as heavy chains. That is, this chain is called as the heavy chain. So same way, 2 heavy chains are present. So we represent it as H2. So if they represent H2L2, this is the representation used for an antibody molecule. Okay. Now coming to the chains. So if you take a light chain, a single light chain, it consists of 2 regions. This shaded portion is the variable region of the light chain and the other one is the constant region. The same way in heavy chain also, the variable regions will be there and then the constant regions are mentioned. So this C here mentioned in our diagram represents that it is a constant region, and the variable regions are represented by the letter N, N terminal end of the chain. So this is the structure of the chains, and both the chains are held together by disulfide bridges. Okay, so all these bridges are the disulfide bridges. If you see, there are only 2 disulfide bridges which are actually joining the heavy chain and light chain. Bridge number 1 and bridge number 2. Okay, so only 2 interchain disulfide bridges are there. Remaining all the bridges are intrachain bridges. Okay, right. So here, now let us solve the question. So they have asked us to label A. A, they have combined the variable regions of both heavy chain as well as light chain. This region is the antigen binding site. So A is the antigen binding site. So antigen binding site will be made up of the variable regions of both light chain as well as heavy chain. And this is our variable region of light chain, constant region of light chain, and the chain they have asked us to label. So this is nothing but the light chain. Right. Now coming to F. So in the question actually only one bond is represented like this. So for our understanding here the entire disulfide bridges are represented. Okay. So this represents the disulfide bridges of the antibody molecules. Disulfide bonds or the disulfide bridges. And lastly, coming to E, chain. This chain is nothing but our heavy chain. So we have labeled all parts of the antibody molecule. And if at all even structure of an antibody molecule is asked, this are the points. These are the points which we have to explain. And since these antibodies are found in the blood, the immune response mediated by the antibodies are called as humoral mediated immunity. Okay, thank you. All the best.